presented by Caltech.
It's my pleasure to introduce Elliot Meyerowitz as tonight's Watson Lecture speaker. Elliot is the George Beadle Professor of Biology and a Howard Hughes Medical Institute investigator at Caltech, where he's been on the faculty since 1980. Prior to joining Caltech, Elliot earned an undergraduate degree in biology from Columbia University and a PhD degree in biology from Yale University. Following his graduate work, Elliot spent time as a postdoctoral fellow at the Stanford University School of Medicine. The Meyerowitz Laboratory studies the development of Arabidopsis thaliana, a small mustard and a widely used plant model system that his laboratory and others popularized beginning in the early 1980s. We'll be hearing more about that tonight in his presentation. From 2000 to 2010, Elliot was chair of the Division of Biology, and in 2011 and 2012, while on leave from Caltech, Elliot served as the inaugural director of the Sainsbury Laboratory at the University of Cambridge, which is focused on elucidating the regulatory systems underlying plant growth and development. Elliot was selected for this position based on his leadership and preeminence as one of the world's leading plant biologists. Elliot is also a past president of the Genetic Society of America, the International Society for Plant Molecular Biology, and the Society for Developmental Biology. Among Elliot's many honors are the Genetic Society of America Medal, the International Prize for Biology of the Japan Society for the Promotion of Science, the Lounsbury Award of the National Academy of Sciences, the R.G. Harrison Prize for the International Society of Developmental Biologists, the Balsan Prize, the Sibthorpe Medal of the University of Oxford, the Dawson Prize for Genetics from the University of Dublin, and most recently, the Gruber Genetics Prize in 2018. And that's actually quite a mouthful. Elliot is an elected member of the U.S. National Academy of Sciences, the American Academy of Arts and Sciences, and the American Philosophical Society. He's also a foreign associate of the Academy of Sciences of France and a foreign member of the Royal Society, one of the world's oldest honorary societies. The title, as you heard of Elliot's talk, is Plant Growth, How Stem Cells Make Stems. Please join me in welcoming Professor Meyerowitz to the podium.
Thank you, Steve. Well, thank you all for coming. I want to talk about how plants grow today. And I'm going to talk about some fundamental questions in developmental biology. But at the beginning, and then again at the end, I'm going to try and relate these to some human problems. So let's begin with the developmental biology question we're trying to answer, which is a question that's of importance for plants and also for animals such as ourselves. The questions are, how do shapes form from stem cells? For example, how does something like our ears take the elaborate shape that they take, or in this case, a flower, which we're showing forming its first sepals from a little round primordium, but then running it backwards so that you can see the complicated beginning of the shape of a flower, running back to the stem cells that make the flower. How do the cells in an organism know to create a shape like this? It's the problem of morphogenesis. The other problem I want to talk about is how do general patterns form in growing plants or in any organism. We're showing here the pattern of leaves around a stem, and you can see that it's very regular. We're all familiar with these phyllotactic patterns, and I'll talk more about those later in the talk. A human cognate of this might be something like the fingers on our hands. We have 5 of them, usually not 6. And they're each of a different type. It's a complicated pattern. How do those cells in our embryos know to turn into a pattern of 5 different types of digits or a spiral pattern of leaves around a stem? And these are fundamental questions in developmental biology. And I will talk about a new discovery that we and others have made in plant biology that shows a new mechanism by which some of these patterns form, and which gives us a possibility for intervening in the architecture of plants in ways that may be of interest and of importance for human welfare. So the answer to this question that I'll give you is that mechanical responses of cells control the developmental patterns. It's long been known that cells signal to each other through chemical substances and that patterns can be created that way, but it's only recently been found that cells equally signal by mechanics, by pushing on each other and pulling on each other in ways that send specific signals through the tissue and give us new ways to intervene in developmental patterns. Before I do that, I want to talk a little bit about why it's important to understand plant growth, the human necessity for more concentration in plant biology. I'm starting, as all scientists do, with a plea for funding in his field, and here's my plea. First of all, there isn't enough food for everybody in the world. There's enough food in the world, but we'll come back to this place, but these places, there's too little food. Not enough is grown. The agricultural productivity is insufficient to feed everybody. And so almost 1 billion people regularly suffer from hunger, and most are women and children. More people die of hunger every year than from AIDS, malaria, and tuberculosis combined, and 1 child dies every 6 seconds from hunger-related causes. So it's an enormous humanitarian issue. Now, these parts of the world have too little food. Some parts of the world can make a great deal more food, but this has been true for centuries, and there's no way to get the excess food from here to here that anybody will pay for. And so what we have to do is learn to control plant growth in ways that we've learned to do in our agricultural fields, but that are suited for the agricultural fields in different sorts of climates. There's another issue with our agricultural productivity. We grow more food than we need. In fact, we have obesity problems, as is well known. But for every calorie of food that goes on your table, American agriculture uses 10 calories of oil. It burns 10 calories of petroleum. We can do that because we're rich, but these people can't do it. And so in addition, it's not sustainable in the long run, and it's not great for the environment because of the excess CO2 that's emitted from these sorts of practices. So even from our sort of agriculture, it would be good to know new ways to breed plants and new ways to intervene in plant growth to create higher agricultural yields in a more sustainable way. Now, it's not only the humanitarian problem of food that's important, but there are also political problems. This is a graph that shows the food price index over the past few decades, and the red lines are revolutions that have started. You may notice a correlation between the revolutions in Syria and in Somalia and in Haiti. They correlate with peaks in food price index. So if we want a world that's peaceful and politically stable, we have to solve this food problem because it's increased prices of food that lead people to riot, turn against their governments, and that make their governments ineffective, demonstrates the ineffectiveness of the government. And so for those reasons, we have to create more food in the appropriate places on Earth. Now that's the picture today. Here's the picture a few years from now. There we are at about 7.5 billion people on Earth right now. But by 2040, there's going to be 9 billion people, 20% more mouths to feed. So the situation is bad now, and it's getting worse. Well, you might say we can plant food on more land, but all the arable land in the world is being used. And in fact, the amount of arable land, 38% of the world's land area in 2000, and down to about 36% now is arable land. The amount of arable land is decreasing because of desertification and because of the expansion of cities in agriculturally rich areas, cities like Shanghai, that are expanding rapidly out into the surrounding farmlands. And so the situation is bad now, and it's going to get worse if we don't better understand how plants grow and use that knowledge to increase our agricultural productivity. It's going to get even worse than that because as climate changes, crop yields go down, and they can go down very fast. Optimal agricultural practices at a 22 degree centigrade country, if there's a 1 degree or 2 degree increase in temperature, which there is going to be as a result of carbon dioxide in the atmosphere, we'll lose 20% of our agricultural productivity. So we're facing a lot of challenges for the future that we have to meet. So let me introduce plant stem cells and why they're of importance for this problem, and some of the methods we use for their study, which I'm going to talk about only very briefly. This is Arabidopsis thaliana, the plant we use in our laboratory. It's not a crop plant, but it's not different in its fundamental processes from any crop plant. This is what it looks like close up, where there's the roots, the leaves, the stem, and flowers at the top. When a seed germinates, it's just a seed, and there's two cotyledons that pop out of the top, and then over time, a collection of leaves are made in a rosette, and then a stem is created, and then flowers are made when it's time for the plant to go into its reproductive phase, and all of these tissues arise from a single small collection of stem cells called a shoot apical meristem, which is present in the embryo, and the cells divide and produce on the sides of this stem cell collection the leaves, and they pour out behind them the cells that will make the stem, so that the shoot apical meristem is pushed ever higher in the plant, so that it's at this point right up here at the tip, and it's making flowers on its sides and still continuing to pour cells out behind it that make the stem go ever higher. This is what a shoot apical meristem looks like, much and large. These are individual cells, and that's the meristem, and it's creating on its flanks in this picture primordia for flowers late in the life of the plant. So there's an early flower primordium, and there's a slightly later one, and there's a slightly later one beginning to make the sepals, whose morphogenesis I showed in the first slide. And so it's this small collection of a few hundred cells that creates the entire above ground part of the plant. So that if we want to understand how plants grow, how they make leaves and flowers and seeds for human consumption, we have to understand how patterns are created and how cells are created in this stem cell population, the shoot apical meristem. Now, to give you an idea of how big that really is, I've taken from the internet a picture of a needle with a thread through it, and that's the Arabidopsis shoot apical meristem at the same size, small enough to go through the eye of a needle. It's not very much different in size from a shoot apical meristem in a giant sequoia tree, which over 1,000 years pours out behind it that gigantic tree. But it's a very small collection of cells. And so that is at the end of every branch and every tree outside on campus. Every time a new leaf is made, a new meristem forms in between that leaf and the stem, and that makes a branch, and when that makes leaves, it makes another branch. And so you get the fractal growth of trees, and at the end of every one of those branches, there's a tiny collection of stem cells, the shoot apical meristem. So let's talk about pattern formation in the shoot apical meristem. Here's our meristem right at the tip of the plant, and there's a picture of it sped up 3,000 times so that we can see it as it's making flowers and as the cells individually in the meristem are dividing, which the astute students in the audience will see all of these cells but one divide in the course of this 65-hour movie. How have we made a picture like this? Well, in brief, what we do is we put a jellyfish protein into the plant's genome, a gene coding for a jellyfish protein that fluoresces in the green. And we direct that protein by adding some extra piece of protein to it to go into the membrane that surrounds each cell in this case. And then we put the meristem in a laser scanning confocal microscope, which scans the laser light of a particular wavelength across the image and focuses it to an exact point and scans it across a single plane in the meristem. We then step the meristem up a bit and do it again, and step it up a bit and do it again. Or in the case of our microscope, step the lens down a bit and do it again, and the lens down a bit and do it again. And then we use a computer to piece all those sections into a three-dimensional image, which we can then create time lapses from by taking images one time after the other. Here's another example of one of these laser scanning confocal microscope pictures of a live meristem in which we've colored differently the proteins from a number of different genes so that we could watch the changes in gene expression patterns that occur as flowers develop. So that's our primary methodology, is the use of a commercial laser scanning confocal microscope with a few additions of our own. A piece of equipment that costs $600,000 or $700,000, but does the job. Now, that's the introduction to the plant stem cells and the methods we use for their study. One of the things that we found in our laboratory is that these stem cells respond to mechanical forces. And I want to show an example of that now, and then I'll loop back to the relevance of this example for the problems I'm talking about at the end of the talk. And then after that, I'll loop back to the relevance of our discoveries for the problems I introduced at the beginning. So what we have here is a shoot apical meristem, and we've poked it on the side with a little piece of glass rod. So remember, that's much smaller than the diameter of a human hair, and these are the individual cells, and the red color represents the concentration of calcium ions in the cytoplasm of these cells. So what we can see is that if we poke a few cells here with a glass rod, those cells respond by immediately increasing their calcium concentration by drawing calcium in from the calcium-rich cell walls that surround them. And then they signal their neighbors, and they signal their neighbors, and they signal their neighbors. So the entire meristem knows that these cells have gotten poked, so that the meristem responds to mechanical forces. Now, the way this works in this particular case, if anyone's curious, is that there are stretch-activated calcium channels in the membranes underneath each cell wall. And the way we know that is that we can take a single cell and remove its wall and put it up against the suction pipette and stretch the membrane by pulling a little vacuum in the pipette, and that activates this exact same calcium response in the cytoplasm here. That's all the vacuole in the middle of this cell. And so that stretch-activated calcium channels draw calcium in from outside the plant to inside, and this is one of the pieces of evidence that these plant cells know that there are mechanical forces being applied to them. I'll come back to this later. Now, here's a view of some plant cells in the shoot apical meristem to talk a little bit more about the mechanical situation of plant tissues to make understandable some of the experiments that I'm going to talk about. This is a plant cell. This, if people in the front row can see the nuclear membranes surrounding the nucleus there. This is the cytoplasm, and this is the cell wall that surrounds each cell. It's about 100 nanometers thick, very thin, but extremely strong. Each of these cells is under about 10 atmospheres of pressure, about 3 times what's found in a bottle of champagne, or what's found in a high-pressure bicycle tire. And this wall resists that pressure so that the pressure persists in each cells, and there are little channels between the cells that let water move between them, so they're all at essentially the same pressure. Very highly inflated. If you break some of these walls, you can get explosive decompression. That's what happens when you bite a carrot and it goes snap. And if you let the carrot dry out a little, so that the turgor pressure is lower, it doesn't do that at all. So we have these cells that are under high pressure, sitting all attached to each other in ways that they can't slide with respect to each other or move with respect to each other. So that's one aspect of the mechanics of a plant tissue, is turgor pressure, and a very strong and resistant cell wall, which has some elastic properties. An increase in turgor will make them expand a little, and a decrease reduce their length a little bit, but also a viscous aspect, so that as the cells grow, the walls deform and grow larger in a way that they won't come back again elastically. So they're viscoelastic resistance to turgor pressure. Now, it's been known since the 1850s that there's one other balance of forces in the mechanics of a plant, which is that the epidermal cells, the cells on the outside of the plant, are under tension relative to the cells inside the plant. So each of the epidermal cells is under high turgor pressure, but in addition to the turgor pressure pushing on their walls, there's an additional stretch applied to their walls because the cells in the middle of the plant have walls that can't quite resist all of their turgor pressure. So there's residual pressure from the internal tissues, which stretches like a balloon skin, the last layer of cells, which gives the final resistance to the mechanical tension and puts the plant into equilibrium. So we have 2 forces at work in the epidermal cells of the plant. One is the turgor pressure in each of the individual cells, and the other is an overall stretch in addition to that, which stresses the whole tissue. And that overall stretch will play a role in some of our experiments. So given this, the engineers in the audience know that because we have an epidermal layer that's under tension and that's thin, that we can do mechanical calculations on this tissue using thin-walled pressure vessel calculations, exactly what engineers use to calculate pressures on a boiler or something of that sort, and so that we can calculate from first principles, based on the shape of the tissue, the stresses in different directions in different parts of the tissue, where stress is force per unit area, which leads to strain, which is expansion if it's sufficient. And so if we have a hemispherical area, like at the top of a meristem, all the stresses are equal, that is, they're isotropic, the same in all directions. In a cylinder, the stresses are anisotropic. There's twice as much stress this way as this way, which is why when a cylinder that's unreinforced blows up from the pressure inside it, it opens with a gap in this direction and not this way, because there's twice as much stress this way. And we can calculate that for our meristem, so that at the top of a meristem, the stresses should be isotropic. Around the stem beneath the meristem, there should be twice as much stress circumferentially as there is up and down. And at a point between a new flower primordium and the meristem, there should be strongly tensional forces across the saddle point and much less tension, potentially even compression, orthogonal to that, at right angles to that, going from the flower into the meristem. So what does this mean to us in our studies of plants? How plant cells sense and respond to tissue shape via mechanics. Now that we know the stress pattern in the epidermis of our tissue, I can reveal to you that the plants also know the stress pattern in their epidermis. And it's this way that each of the cells has in it a set of proteins that are long filamentous proteins called microtubules that are in a helical array just underneath the plant's outer membrane and just inside and attached to the cell wall through proteins that go through the membrane. And these so-called microtubules, which were first discovered in plants in the 1960s, but also exist in our cells, respond exactly to these tissue stresses, so that when the stress is anisotropic, it's greater in one direction than another, the microtubules align in the same direction as the stress. And so by looking at the microtubules in a plant tissue, we can see that the cells know the stress pattern in which they sit, the tensor field of the stresses in which they sit, so that the microtubules are in no particular orientation in the isotropic parts of the meristem. And indeed, they change their orientation all the time. Every hour, they're aimed a different way. But if we look in the saddle point between a flower primordium and the meristem, the microtubules are strongly aligned over the saddle point, and around the circumference, they're aligned around. So here are the microtubules, which people might be able to see, these long lines that we fluorescently labeled and put into our laser scanning confocal microscope. And you can see at the top of the meristem, they're in no particular orientation, but across the saddle point up here, they're very strongly aligned with the principal maximal direction of anisotropic stress. So the readouts of stress pattern and another demonstration that the plants are responsive to their mechanical state. Now, how do we know that it's stress and not some other thing that's aligning them, including strain for the engineers who are already wondering about this? And I won't go into how we know that it's stress and not strain, but what we've done here is made a little tiny vise and squeezed the meristem back and forth. And we squeeze it in, the microtubules all realign to the new principal maximal direction of anisotropic stress. I just love that. Before and after, and we release it, it goes back again. So we can change this pattern by externally changing the stress pattern. There's another way we do this in the laboratory because it's easier than putting a little vise around the meristem, which is we use a laser and we blow a hole through the epidermis in the meristem. A little dye-coupled laser hooked to our microscope. So this is a finite element method model showing the principal directions of stress, which equal the principal directions of the microtubules in a computational model of this tissue. And if we poke a hole in the middle, because the overall tissue is under tension, it tends to pull the hole larger. And when it pulls the hole larger, that creates circumferential stress around the site of the laser ablation, and the microtubules should realign to that stress. And in fact, they do, which we can see here in one of our movies where we've used the laser, and the red shows where we've popped the cells, and you can watch the microtubules around there align from a random pattern to a pattern that's purely circumferential to the hole that we've created. Actually, we didn't use a laser for this particular experiment. We used a needle. It does the same thing. And so we can change the stress pattern and get a predictable change in the pattern of these subcellular structures, which tells us that the plant is responsive to the mechanical stress pattern. So that while most people can't calculate second-order tensor fields, Caltech undergraduates and shoot apical meristems can. And there's the proof. Now, why is that important for the plant? And the answer has to do with the cell wall. Now, one of the predominant components of a cell wall, as everyone knows, is cellulose, which is a long, fibrous carbohydrate, which is extremely stiff. You pull on it and it doesn't expand. It's about half as stiff as steel. And it's laid out in a pattern in the cell wall so that if a cell, for example, is a long cylindrical cell and the cellulose is wrapped around it like this, it's so stiff that it reinforces that cell like a barrel, like barrel hoops, and when the cell expands, it expands at right angles to where the cellulose pattern is. Now, how does the cellulose get into a particular pattern in the cell wall? And the answer to that is that the cellulose synthase complexes are attached to these microtubules, and they run along the microtubules and project out through the plant membrane and into the wall this carbohydrate fiber, so that the direction the cellulose fibrils are in the plant cell wall is dictated by the direction in which the microtubules are aligned, which in turn is dictated by the stress pattern in the tissue, all with delays, which are important for the models, but not something we have to think about. And so that when a plant tissue like a meristem epidermis is under tension in a particular direction, it gets reinforced by cellulose against that tension, and the cells then start to expand opposite to the tension because they're reinforced against expansion in the direction where the tension is. And so the shape of the tissue creates a stress pattern. That stress pattern aligns the microtubules. The microtubules cause reinforcement of the cell wall in an anisotropic fashion, and that reinforcement causes a future change in the shape of the tissue, which then changes the stress pattern, so that it's a constant race towards catastrophe by an epidermis of a shoot apical meristem, but it's always resisting catastrophe by reinforcing itself in the direction it's about to break or burst, and that causes future shape change, which causes future shape change, which causes future shape change, so that the responses to the mechanical stress that I've talked about create the morphogenesis of the plant. Here's some evidence that that's the case. This is a field emission scanning electron microscope picture of the cellulose fibrils in meristem cell walls. So this is one of these meristem epidermal cells. There's the top, where you can see at the top of the meristem where the stresses are isotropic, the cellulose fibrils are in all directions. But on the sides of the cells, since the turgor pressure is pushing the top up against the free space, then there's strong tension parallel on the side of those walls, and you can see the alignment of the cellulose fibrils there. So that observations fit our predictions. And one other prediction of a model of morphogenesis like this is that if we prevent this mechanism from working, morphogenesis should go wrong. And that's shown here, where we've depolymerized the microtubules using a drug that's used for cancer treatments in humans. And over time, the meristem just flattens out and none of the shapes occur. And so that the shape of the tissue is a response to mechanical stress through this mechanism we've identified. Now, there's one more thing that's also a response to mechanical stress in this tissue. There's more than one thing, and I will talk about a few of them. But here's one more to add, which is the pattern of cell division. When a meristem is growing and providing the redwood tree behind it, the cells in the meristem are dividing ever so slowly. They divide once every 12 or 24 hours. But over 1,000 years, that can produce a considerable number of cells. Those cells divide in a particular pattern. Every time they divide, they're expanding in one direction, and then a new wall is created to create two cells. So how do the cells know what plane to put the new wall in? And the answer to that is also mechanical stress. Because what happens in the division of a plant cell is that this microtubule array, which before occupied all of the cell, collapses down from the direction in which it's wound to make what's called a preprophase band, and that marks the site of the new cell wall. And so that when we do our ablation experiment, we poke a needle into this meristem, and we wait 24 hours, you can see that all of the new cell divisions are now parallel to the new stress pattern. And so that the morphogenesis of the tissue depends on the stress pattern, and this cytoskeletal microtubule response to the stress pattern, and also the planes in which the cells divide depends on the stress pattern. which also changes the stress pattern because a new wall reinforces the tissue against stress by adding more material. And that in turn changes the future pattern of cell division and the future shape of the tissue. So all of these things are happening at once, but it defines the mechanism by which the plant creates its new shapes. So here's our first conclusion, and there will be a second conclusion, and then I'll stop. Our first one is that stress is generated by tissue shape and an epidermis in tension. This mechanical stress controls the microtubules and thus the cellulose direction in the walls. The cellulose controls subsequent cell expansion, which in turn changes the stress pattern, and it also controls subsequent cell division, which in turn changes the stress pattern. And so it's a novel developmental mechanism which shows that tissue shape is sensed locally by cells, which leads to future changes in tissue shape. The physicist in the audience will recognize a similarity of this to Einstein's theory of gravitation, which Wheeler once summarized by saying that gravity bends space and space creates gravity. It's very similar. And indeed, the space-time universe and the stress-strain pattern in a meristem are both fourth-order tensor fields. Okay, let's talk about one other aspect of this stress response in growing plants, how mechanical forces create now the phyllotactic pattern, the pattern of flowers around a stem. Now, phyllotactic pattern is very familiar to all of us, that most plants have a spiral pattern in which they make one leaf or one flower after the other with a radial angle that's about 140 degrees from one to the next, to the next, to the next, to the next, to the next. And we can follow this in a pine cone or a sunflower or a piece of broccoli or anything else. And the question I want to answer now is how this meristem, this round collection of cells, knows to make a new flower here, and then the next flower there, and then the next one there. How does it measure these radial angles from what's generally a symmetric-looking tissue? And this is a problem that has bedeviled mathematically inclined biologists for a very long time. The ancient Greeks talked about spiral patterns in growing roses. Theophrastus, who's a student of Aristotle's. In the 1860s, Wilhelm Hofmeister, who was the third greatest botanist of the 19th century, Darwin and Mendel, made the first causal model of phylotaxis. And Turing, who we're all familiar with as the inventor of the concept of computers and important in breaking codes during World War II, and about whom there's a recent movie, when he committed suicide, the problem on which he was working was a mathematical description of phyllotactic pattern and attempting to find the mechanism by which plants could make these lovely patterns. Well, here's the answer, at least so far as we know it. Here's in Arabidopsis phyllotactic pattern. So there's our shoot apical meristem right down in the middle there, and surrounding it is all the flowers that the meristem has made over the past week or two. And so we can see that we go from an older flower to a next younger, and we can go right through in that phyllotactic pattern with each new primordium at an angle of 130 or 140 or so degrees from the previous one. You'll remember from school that this is supposed to have something to do with Fibonacci numbers. Doesn't? Not everything you learn in school is right. So what is I'm being facile here. It has something to do with Fibonacci numbers, but they have nothing to do with a plant. It has to do with the way your eye perceives these patterns. In any event, how is it that flowers know to form at the right generative angle, one after the other? And the answer, which has been known in part since the 1930s, is that there's a plant hormone called Auxin, indole 3-acetic acid, which was first discovered in 1926 by Fritz Wendt, who was after that a Caltech professor. And that hormone, shown by this green reporter for hormone concentration that we've created, or that we've borrowed from somebody and put in our confocal microscope, reaches high concentration at those places where the next few flowers will be. And we know that that's the cause of the appearance of the new flower, because in the 1930s, Robin and Mary Snow put little dabs of Auxin on shoot apical meristems and showed that where they put the dab, they could invoke a new leaf or a new flower. So this plant hormone causes gene expression changes that lead to the creation of a new flower. And our question becomes, how does this plant hormone get to high concentrations at this particular angles in the meristem and not any other concentration? And the answer turns out to be the transport of the hormone, not its local synthesis. And so I have to introduce a few things about auxin, which is central to all understanding of plant development. So auxin, as I said, induces new leaves and flowers. One other thing it does, one of its most early effects when you put it on a shoot cell, is that it causes the wall to weaken by activating some pumps that pump protons out of the cell, making the wall more acidic, which weakens it. And so a cell that's high in auxin starts to expand a little bit because the wall is weakened and it can't completely resist the turgor pressure and the tissue tension that comes from the underlying cells. And so cell expansion is a result of increased auxin concentration. Now, auxin is a weak acid. It has a pK for the chemists of a little under 5, and so that at a pH of 7 in the cytoplasm of a cell, it's an ion. Its proton isn't attached. It's a charged molecule, and that charged molecule, indole acetate, will not go through the membrane that surrounds the cell. So auxin is trapped in cells and can't get out. It is let out by a protein called an efflux carrier. And if the efflux carrier is in the membrane, then the auxin can get out. And when it gets out, because of these proton pumps, the pH of the cell wall is low, 5.5, and so much of the auxin becomes reassociated with a proton. It's no longer charged, and it can diffuse through membranes without any carrier, although there are such carriers, it can do it without them, and it can diffuse back into the cell where it originated or diffuse into the cell adjacent. Now, if the efflux carriers are found only in the bottom of each cell in a line of cells like this, the auxin can only get out at the bottom of the cell. Once it's out there, it can go back into this cell, but then it can only come back out the bottom again. If it gets into this cell, it can't come out here because there's no efflux carrier. It only goes down to the bottom and comes out there. So the asymmetric distribution of the efflux carrier in the membrane of the cell creates a net direction of auxin flow. So this hormone has a private circulatory system, and it can move in its own ways through a tissue that depend on the local positioning of the efflux carrier in the membrane. Here's one proof that the flower development is a result of that, and this is a mutation found by Kiyotaka Okada in Japan many years ago, which knocks out the activity of the efflux carrier that's important in the flowering shoot apical meristem. And when you knock that out, you don't get any flowers. Hence, the gene that codes for this protein is called pinformed. And so there's a pinformed, that's the meristem, and no flowers get made because auxin can't flow around in the tissue. So this tells us that it's the transport of auxin through this pinformed efflux carrier that creates the new locations where flowers are formed in the shoot apical meristem. Now there's another aspect of pinformed 1 that I want to mention before I show the next slide, which is that I pointed out before that one of the other effects of auxin, besides weakening cell walls and besides being trapped in cells and only allowed out by this efflux carrier, is that it changes patterns of gene expression. And one of the genes whose expression is changed by auxin is the efflux carrier gene. So a cell that's high in auxin has more of the efflux carrier because the gene is induced, and after a half an hour, there's more of the protein in the cell, which creates a conundrum for us when we want to think intuitively about what a cell is doing in a shoot apical meristem. Because if I'm a cell that's high in auxin, the first thing I do is I induce my efflux carrier protein, and I spit my auxin out to my neighbors. So now I'm a cell that's low in auxin. My neighbors now have picked up all this auxin because it's out in the cell wall and it diffuses back into them. It changes their pattern of gene expression by causing them to induce their auxin efflux carrier, and they spit their auxin out to their neighbors. So they become a high auxin cell, but then they become a low auxin cell, and some of it comes back to me, so I become a high auxin cell again. So there's an interplay of auxin concentrations and auxin efflux in the meristem that we'll come back to in a moment, but I want to show you one effect of that at first, which is this movie, which shows us the auxin efflux carrier in the plasma membranes of these cells in a movie, and the places where it's brighter are the places where the auxin concentration is higher, because more of the protein is made in cells that are high in auxin. And you can see that it's a very dynamic pattern as the cells divide. There's a flower primordium beginning to form with its high auxin, but after a while it becomes low in auxin because the auxin goes to the position where there's a new primordium, like over there, but then after a while it comes back out there and goes back into the meristem because of this dynamic interplay of auxin in and out of cells. And so we can look at the patterns of auxin concentration, which are highly dynamic, by looking at PIN1 protein, but we can also look at a little higher magnification where our PIN1 protein is staying green and the wall between the cells, or in this case, the red is the membrane between the cells, and you can see on which side of each cell the PIN1 protein is, so that that cell is sending auxin into that cell, and that cell is sending auxin into that cell, because green is the efflux carrier and red is the barrier between the cells. And so you can tell the direction of auxin flow by looking carefully in our microscope at what's going on. And when you do that, we've drawn arrows here for all these directions. You can see that at first the auxin moves in towards a new primordium, and then after a while it moves back out again. So why is that, and why does it happen in a particular pattern? Well, the rule that we can see in these micrographs is that the auxin from one cell generally is pumped toward the neighboring cell that has more auxin. So that each cell seems to know the auxin concentration of all of its neighbors, and when it's high in auxin, it makes efflux carrier, and it puts that efflux carrier on its side that's adjacent to its highest auxin neighbor. And so it's a positive reinforcement of the auxin in the neighbor, but of course the neighbor then builds up its efflux carrier and sends the auxin back in other directions. And so the rule is that auxin is being pumped up the gradient by the efflux carrier, the energy for which comes from this pH difference, which is created by the proton pumps that burn ATP for the experts. And so here's what we know from our observations is that locally high auxin concentration causes new primordia, and it gets high locally by transport and diffusion. That auxin efflux carrier moves the auxin, and its gene is auxin-induced, so the rate of transport from a cell depends on the auxin level in the cell. And then the auxin efflux carrier is polarized in cells and points towards neighboring cells with the highest auxin concentration. So let me ask you, is that now a sufficient explanation for the phyllotactic pattern? The high school students are in front. I want an answer. Can you see the whole thing in your head? If you can come work in my lab, but I can't. No one can. So what we had to do is we have to translate these three statements into the language that our plants speak, which luckily for us is also the language that Caltech undergraduates and that computers speak. And here are those three statements translated into a set of ordinary differential equations, so that there's the concentration of our PIN1 efflux carrier with time in cell I is a function of creation times the auxin concentration minus degradation, which is first order in PI concentration. There's the concentration of auxin in cell I, which equals production minus degradation minus transport out plus diffusion back in. And there's the direction from auxin transport, where the PIN1 is pointed from cell I to J, which is proportional to the amount of auxin transport in that direction is proportional to the amount of the auxin efflux carrier in cell I times the amount of auxin in cell J, because it's the adjacent cell to which it's pointing. And if we take these equations and we solve them using our laptops in a hexagonal array of artificial cells in a plane, there it is. There's our phyllotactic pattern. So yes, that dynamism with appropriate movement of auxin is exactly what it takes to make a spiral phyllotactic pattern, and every other phyllotactic pattern if we change the relative size of the meristem to the rate of auxin transport and diffusion. And so that's a simplified model, of course, but it shows us that we might be on the right track. There's the real model, which maybe is slightly past that. Maybe the high school seniors can get this one. but the juniors have only taken 1 year of calculus, in which we've added in the Auxin concentration in the wall between cells I and J, which is critical for understanding the diffusion laterally as well as the diffusion back into the cell of origin. In that last model, I only had Auxin going from one cell to its neighbor, but of course it goes out into the wall and then can go back in either direction, and that's what's included in this model. And we can also make a more realistic mechanical substrate for our meristem, where the cells aren't in a hexagonal array, but have the number of sides that real cells and real meristems do. And in that last array, I was adding one new cell in the middle each time to create a changing field, but this time we've got all the cells dividing just in the patterns in which they do divide. And we get exactly the picture that we saw when we looked at the real Auxin concentrations in that earlier movie. And so those rules are sufficient to explain the phyllotactic pattern. And it has to do with the dynamics of Auxin transport. Now, where does the mechanics come into this? And it's like this. We have local high Auxin concentration. This is our rule 1, causes new primordia, and it gets high locally by transport and diffusion. Our rule 2 in our model is that Auxin efflux carrier moves Auxin, and its gene is Auxin induced, so the rate of transport from a cell depends on the Auxin level in the cell. But our rule 3 was a little bit tricky, and probably some people in the audience noticed that. Here's our rule 3, which is the Auxin efflux carrier is polarized in cells and points toward neighboring cells with the highest Auxin concentration. Now, how does a cell know the Auxin concentration of its neighbors? Auxin is sensed in a cell by a receptor for Auxin to which Auxin binds, which then activates genes in the nucleus of the cell. So every cell knows its own Auxin concentration, but how can a cell know the Auxin concentration of its neighbors? And that's where the mechanics comes in, because what we've shown is the following. Now, here's a picture. Let me just give you another picture of the mechanics of the tissue, and then we'll talk about the answer. So these are now nuclei that we've labeled in one of our movies. And you can watch the nuclei moving as the cells divide, and you can see that the cell divisions in the developing flowers are much faster than they are in the meristem, but you can still see cells dividing in the meristem. Now, if we take a computer and we look at the positions of all these nuclei, we can follow them, eliminating the ones that have divided, so we use the others as fiducial markers, and it shows us how the cells are expanding in the meristem as it grows, because they're pushing the nuclei apart as they expand. And then we can make a heat map of that, and what we can see is just where the Auxin concentration is highest, as expected, the nuclei are moving apart from each other more rapidly. Because as I said before, when we have a high Auxin concentration in cells, it weakens the wall and those cells expand more than their neighbors. And we can see that expansion is real because orange means faster movement of the nuclei apart from each other and blue slower. Now think back to what I said about the tissue and its cell walls. This is a tissue under a lot of mechanical tension. And if one cell starts to expand, that means it's putting a lot of stress on its non-expanding neighbor, because the neighbor is trying not to expand, and so you have a huge increase in the stress in the shared wall when Auxin concentration goes up in a region of the meristem. And it's this mechanical stress that causes the Auxin efflux carrier to go to the right part of the plasma membrane. And the evidence for that is one experiment here where we've poked our hole. We have a computer model that indicates that once we poke the hole, the highest stresses in the walls are where it's red here, and the differential between the stress in this cell and its neighbor is greatest. So if we actually laser ablate 2 cells there, and red is our Auxin efflux carrier, and green is our microtubules now aligned around our hole, newly aligned circular to the hole we've created, and the Auxin efflux carrier goes exactly to the most stressed walls. And so it appears that this protein, which is moved around within the cell by little membrane vesicles that fuse with the wall and then suck the protein out of the membrane and move it to other locations, it looks like the net vesicle traffic is towards the membrane that's adjacent to the most stressed wall. So again, we have mechanics, but this time the mechanics is not due to the shape of the tissue and our thin-walled pressure vessel calculations, but the mechanics is due to local expansion of cells. which creates even greater stresses than those created by the shape of the tissue and causes the auxin to be redirected to particular points and create new flower primordia. Here's a movie of a model and a movie of a real experiment, which we've ablated these 3 cells with a laser, and then you can watch the auxin efflux carrier move back to the back of these cells and then to the back of these cells, exactly in the pattern predicted by the stresses. So we have a correlation of the stress pattern with the position of the auxin efflux carrier, and when we change the stress pattern, we have a causal relation between the change in the pattern and the movement of the auxin efflux carrier. So we can come to our second conclusion, which is that auxin causes cell expansion. The resulting mechanical stress controls auxin transport from neighboring cells. New auxin peaks changes which cells are expanding. So again, it's fully dynamic, thereby changing the auxin flow again. So we have mechanically regulated transport of a chemical substance, which is called a morphogen, because it creates new primordia or organs in the place where it goes. And in fact, it was Turing who invented that word morphogen for the thing he was trying to use to explain the phyllotactic pattern, and he came up with a model for it, his so-called reaction diffusion model, which turns out not to be what's going on in phyllotactic pattern, but nonetheless has been a really fundamental process in the understanding of parts of animal development. So let's come back to the first mechanical response I showed you. Here's our calcium response when we poke a cell. A third mechanical response, perhaps, and we have computational models of this too, so that you can see just what's happening. We first poke the cells, and then they tell their neighbors they've been poked, and we can exactly replicate the waveforms of this thing. What relevance does this have for what I've just talked about? And the answer is that it's extremely relevant to the auxin efflux carrier. Because if we prevent the increase in calcium by adding a drug that blocks the calcium, stretch-activated calcium channels, the pin 1 doesn't move. It stays absolutely where it is no matter what we do to the stress of the tissue. So the first step in mechanical sensing that's important for the movement of the efflux carrier and therefore for the phyllotactic pattern is an increase in cytoplasmic calcium. And if we wash the calcium out and wait a few hours, well, then the pin 1 responds to the hole just as it would had we not had any treatment at all. So it's a reversible treatment. So this mechanical response regulates, or is the first step in the movement of the auxin efflux carrier to the appropriate side of the cell so that it can send its auxin to its most auxin-rich neighbor. Now, this same blocking agent does nothing to the microtubules. That's a completely separate mechanism. So we have at least 2 fully separate mechanisms which interplay with each other, though, because the shape of the tissue affects the stress pattern and local expansion affects the stress pattern. And so these 2 separate ways of detecting mechanical stress interplay through changes in shape and changes in cell expansion and changes in cell division patterns with each other, because they're always sensing the stress in different ways, but each of them also changes the stress in different ways. Now, it might have occurred to everyone in the audience that the phyllotactic pattern doesn't depend on poking a meristem with a glass rod. And so I've been invoking this mechanical response, this calcium response, with a glass rod. But what happens in a real meristem when somebody's not there poking it? And the answer is that the cells respond to their own stress changes over time. And this is our calcium response in a living, growing meristem, sped up about 40 times, so that these cells are twinkling all the time in response to everything that's going on in the tissue. So it gives us another readout of changing patterns of stress, we think in this case, in the tissue, and also shows the stress patterns that are changing as the auxin is moving, as the tissue shape is changing. So that overall, our conclusions are, and I'm not quite done after this either, that the cytoskeleton and therefore the cell wall anisotropy, which changes the direction of cell expansion, is controlled by physical force. The cytoskeleton and cell division planes are controlled by physical force. Auxin flow pattern, and therefore the phyllotactic pattern, is controlled by physical force. And cytoplasmic calcium ion levels, and therefore auxin flow, are controlled by physical force. So all of these effects feed back on stress patterns, so all of them affect each other. And the reason we use our computational models is because you can't keep this all in your head. But we can model it in a computer and get back to predictive ways to change the shapes of meristems, the pattern of organs around the meristem, and the cell division patterns, and therefore the way in which the plant grows. And just one demonstration of that is, what does this knowledge give us that might be of use to us in agriculture or in further steps in developmental biology? Well, one clear realization we've had as a result of understanding all this interplay of mechanics between cells is that the cell walls are bearing all the load in this tissue, and so that if we intervene with the cell walls, we should be able to change the way a plant grows, something that would have been unsuspected before understanding the role mechanics plays in developmental pattern formation. And so here's one just example of that. What we've done is we've slightly changed the expression pattern of a gene that codes for a protein that goes out in the cell wall and resplices carbohydrate change. It's a xylan endoglucosyl transferase. And if we just overexpress that a little bit in the epidermal layer cells, there's a normal meristem, and there's the meristem where we've just slightly changed the properties of the cell wall. You can see that the phyllotactic pattern is completely different, and the cell division patterns are different such that the cells are bigger. So we've learned from all this that we can intervene in the growth of a plant in a way that previously we didn't suspect, which is by intervening in the enzymology of the cell wall, which is understood at a considerable level of depth, though not a full level of depth. So it gives us a toolkit now that we can use to redesign plants. Why is that important? Well, here's an example of one recent paper by Steve Long and one of his collaborators at the University of Illinois, where what they did is they modeled the growth of soybean plants. And they calculated how the plants receive sun, reflect sun, and so on. And by optimizing the growth of the plant in their computer model, they could say they could get a crop with 8.5% more productivity and using 13% less water and reflecting 34% more radiation back into space by breeding for slightly different leaf distribution, angles, and reflectivity. Well, now we know how to control this, at least the leaf angles and distribution. And so that their models of crop growth and our models of cell-cell interaction and meristems come together to give us a toolkit that we can now use to alter plant growth in entirely new ways and possibly to answer some of these daunting human problems that result from our failure to fully understand how plants grow. So with all that, I want to thank the people who did this work, and this is a recent picture of my lab, but by no means everybody in the lab who participated in this work. And I'll particularly point out Paul and Tyler, who are sitting in front with the high school students from South Pasadena High School and took them to dinner today, and the rest of the people. Ting was particularly involved in the calcium work, as was Eldad. Paul has worked on meristem chemical interactions. I would even say what Tyler's working on because it's radically different from what I talk about, but in the topic for my next Watson lecture. Pauline did some of the calcium work on the single cells, and so on. I don't want to call out everybody, but of course it's this brilliant collection of students and postdocs who have done all of the work and that have enabled me to stand up here and talk to you about it. So that's all. We won't have formal questions and answers, but I'm happy to answer questions for anybody who wants to come up for a few minutes after the lecture.